Good afternoon and welcome to the Saniona R&D Day, leveraging ion channel targeting expertise for rare diseases. At this time, all attendees are in a listen-only mode. A question and answer session will follow the formal presentations. If you would like to submit a question, you may do so by using the Q&A text box at the bottom of the webcast player or by emailing your questions to questions@lifesciadvisors.com. As a reminder, this call will be recorded and a replay will be made available on the Saniona website following the conclusion of the event. I would now like to turn the call over to your host, Rami Levine, President and Chief Executive Officer at Saniona. Please go ahead, sir.
Thank you very much, Sarah. Good morning and good afternoon to all participants, wherever you are. I'm Rami Levine. I'm the President and CEO of Saniona, and welcome to Saniona's first ever R&D Day. Today, you will hear a lot about the fascinating world of ion channels. You will hear about what we are focused on here at Saniona in our research organization, and you will even get to meet some of our scientists. So without further ado, let's just dive straight into our R&D Day. You have here in front of you our agenda for today. I will start with giving a short introduction about the company for those of you who are not that familiar with Saniona. Right after me, we have our guest speaker, Heike Wolf from UC Davis School of Medicine, who will talk about the untapped potential of ion channels. Jürgen Dreyer, our CSO and founder, will talk about our ion channel drug discovery engine. David Brown, our Director of Discovery Projects and Medicinal Chemistry, will talk about IONBASE and artificial intelligence at Saniona. Palle Christopherson, our Senior Vice President of Research, will do a preclinical review of SAN903. Karen Nielsen, our Vice President of In Vivo Translational Pharmacology, will do a preclinical review of SAN711. And then Rudy Baumgartner, our Chief Medical Officer and Head of Clinical Development, will talk about our clinical preparations for our phase 1 study with SAN711, which will be starting very soon, as well as a review and an update on Tesomet, our lead asset. I will then conclude the meeting before we open it up for a Q&A session. So before we start, just a forward-looking statement. I'm not going to go into details, but I'll just leave it here for a second and dive straight into Saniona and who we are. So Saniona is a clinical stage biopharmaceutical company. We are focused on discovering, developing, and ultimately commercializing our innovative treatments for rare diseases. Saniona is publicly traded on NASDAQ in Stockholm. Our research organization is based in Copenhagen, Denmark, and our corporate offices are being built in the Boston area. Our lead asset is Tesomet, which we are developing for 2 rare disorders. The first one is hypothalamic obesity, and the second one is Prader-Willi syndrome. In hypothalamic obesity, we conducted a phase 2 study, which we presented the top-line results of the blinded phase of that study back in April of last year. We then presented the open label extension of that study in November of last year. And we've been actively in discussions with the FDA to align on the next steps in our clinical development. And we have aligned on a phase 2B study, which we expect to initiate in the second half of this year. Our second indication is Prader-Willi syndrome. Similarly to HO, we conducted a phase 2 study. We have been in dialogue and discussions with the FDA. And similarly, we've aligned on the next step in our clinical development, which will also be a phase 2B study, which we plan to initiate also in the second half of this year. But we are not a one-trick pony, if you will. We are not just about Tesomet. We actually have a proprietary ion channel discovery engine, which in fact we will be focusing most of today on. Our lead asset coming from that drug discovery engine is SAN711, which we are exploring for rare neuropathic disorders. SAN711 is ready to move into phase 1, which we plan to do over the next month and a half, 2 months. SAN903 is about a year behind SAN711. We are exploring SAN903 for rare inflammatory and fibrotic disorders. SAN903 will move into phase 1 in the first half of 2022. And then behind those lead assets, we have a whole library of over 20,000 proprietary ion channel modifiers. And our goal will be to investigate that library with the goal of identifying the next treatment for the next rare disease. Those two first pillars that I just touched on, Tesomet and our ion channel drug discovery engine, is really where the company's focus is moving forward. But having a drug discovery engine also allows us to out-license partner some of our molecules that we don't intend to develop ourselves. And some of the examples that we have already in place of such agreements, the first one is tesofensine for obesity. We don't plan to develop tesofensine for obesity. We partnered with a company called Medix, who have obesity clinics in Mexico and Argentina. They conducted a phase 3 in obesity back in 2019. They submitted the file for approval in Mexico in December of that year, and they are waiting now for approval from the Mexican authority, which we are told will happen very soon. Once that is approved, Saniona will be getting double-digit royalties on sales, and they will then pursue an approval in Argentina as well. Another out-license agreement that Saniona has is for CAD 1883 for movement disorders, which we out-licensed ultimately to Cadent Therapeutics. Back in January this year, Cadent Therapeutics was acquired by Novartis, and under that acquisition, we received an upfront payment of nearly $3 million, and we will be receiving additional milestone payments throughout the development and royalties if and when the product reaches the market. And last but not least, we have an out-license agreement with Boehringer Ingelheim for their schizophrenia program. Similarly, under that agreement, we will be receiving milestone payments throughout the development and ultimately royalties if and when the product reaches the market. Saniona is well funded into the second half of next year, and that is predominantly due to a raise of $65 million, which we announced in August last year. That was a transformational raise for the company and really for the first time allowed us to advance our clinical development, to advance our preclinical assets into clinical development, and to build the organization and hire the expertise that we need in-house to be able to build a fully integrated pharmaceutical company. So we are very excited with the raise, but we are equally excited with the strong institutional support that we have following that raise. That deal was led by RE Capital. There were also Pontifax, New Leaf, and a whole list of other institutional investors. So that's Saniona in a nutshell. But without further ado, let's really dive into the main reason we are here today. And it is really my great pleasure to introduce our guest speaker, Dr. Heike Wolf from the University of California, Davis. Dr. Wolf obtained her PhD in medicinal chemistry in 1998 from Kiel, Germany. In 1999, Dr. Wolf joined the Department of Physiology and Biophysics at the University of California, Irvine. She then became an assistant professor at the University of California, Davis in 2003, and was then tenured in 2008. Dr. Wolf is now a professor of pharmacology at UC Davis. Dr. Wolf's research is focused on potassium channel pharmacology and the design of new ion channel modulating drugs, and has authored over 175 peer-reviewed publications. Dr. Wolf, it's a pleasure having you here with us today. The floor is yours, please.
Rami, thank you for this very kind introduction. For those of you who wonder where Davis is one of the 9 universities of California. It is 80 miles inland from San Francisco, and our claims to fame are the #1 ranked veterinary school in the world and genetically modified strawberries. But let's talk about ion channels. Ion channels constitute excellent drug targets. Ion channels control membrane potential, action potential firing, calcium signaling, and any type of cellular activation and proliferation. So something is going to happen when you modulate ion channels. Nature knows this. The venoms of hunting animals like snakes, scorpions, or these fish harpooning cone snails are full of ion channel blocking peptides. Similarly, many poisonous plants contain ion channel modulators. There are many ion channels in the human genome. There actually is the 3rd largest group of signaling molecules after tyrosine kinases in GPCRs. So we have lots of targets with the opportunity to make some very selective drugs. Ion channels are already safely used as targets for local anesthesia, hypertension, anxiety, epilepsy, type 2 diabetes, and pain. So they constitute well-validated targets. I've included an example table here that is listing some of the ion channel modulators on the essential medicine list of the World Health Organization. I just want to point out nifedipine, diazepam, which is Valium, and lidocaine. And without ion channel modulators, we probably could not feed and clothe the world, because many insecticides target ion channels. Nicotine and the neonicotinoids act on nicotinic acetylcholine receptors. The polychlorocycloalkanes and the fipronils block GABA-A channels, and the pyrethroids keep voltage-gated sodium channels open. So what I'm trying to say here is that ion channels are highly druggable. But if you talk to people in pharma, especially to medicinal chemists, they regard ion channels as difficult drug targets. It is very hard to achieve selectivity in families with a high degree of homology. Ion channels are large multi-subunit membrane proteins. So despite all the advances with cryo-EM, we still only have very few crystal structures in this field. And until last year, we didn't have any co-crystals of an ion channel with a small molecule. And this is what medicinal chemists really want to do structure-based drug design, because they're used to this from the kinase programs. So they really don't like ion channels. In addition to this, the screening assays are technically demanding and typically require electrophysiology, at least in the later stages. You also have to deal with what is called state dependence. So what is state dependence? What you see here is a simple cartoon of a voltage-gated sodium channel. In the closed state, the inner gate is closed and the voltage sensor is in the so-called down position. Membrane depolarization will move the voltage sensor out of the membrane and open the channel. The channel will then inactivate by closing the gate, but the voltage sensors are still up and have to be retracted before the channel can open again. State dependence means your drug is preferentially or exclusively binding to one state of the gating cycle. And this was really just a simple two-dimensional view. A trimeric, tetrameric, or pentameric ion channel with a voltage sensor and/or multiple ligand binding domains will typically undergo many conformations during its gating cycle. So the question is, which one is your drug binding to? And this is really more than an academic or philosophical question, because it will result in voltage-dependent block, meaning practically that your IC50 values, your potencies, change with the assay conditions. What you see here is a classic literature example showing the concentration response curve of mibefradil at different holding potentials in the electrophysiology assay. At a hyperpolarized potential, you have an IC50 of 1 micromole. At a more depolarized potential, the drug is 10 times more potent. And this becomes even more striking as you go across assay platforms, as you move from flux assays to membrane potential sensitive dyes to electrophysiology. And this is something that really confuses medicinal chemists. So you need to interpret it to them. So how can you successfully target an ion channel with all these problems? First of all, you should really understand the science, the physiology and the pathophysiology of the particular channel. You want to choose your targets wisely. And here KCS 3.1 is a really good choice because it's alone in its family. You want to screen smart ion channel focused libraries, and you want to develop deep expertise in electrophysiology in order to deal with state dependence and provide proper feedback to your chemists. And these are all things that I believe Saniona has done well. For the last 2 minutes, I would like to focus on my favorite ion channel, KCS3.1. As you can see, KCS3.1 has many possible binding sites for drugs. I'm going to concentrate on the inner pore binding site, where you see this blue molecule. The first generation of inner pore blockers, like clotrimazole, were very unselective and also cross-reacted to P450-dependent enzymes. The second generation of compounds, TREM34, Saniona, and NS6180, were much more selective, and they have been used to validate the channel as a target for atherosclerosis, chronic transplant rejection, various types of fibrosis, inflammatory bowel disease, stroke, Alzheimer's disease, and asthma. Out of these compounds, Saniona even advanced into phase 3 clinical trials for sickle cell anemia. It unfortunately failed because it did not meet the set clinical endpoint. But it was safe, so it was deposited in the NCATS library, which made it available for investigator-initiated clinical trials. Since we had done animal work at UC Davis, suggesting that KCS3.1 could be a target for Alzheimer's disease, the UC Davis Alzheimer's Disease Center had planned a clinical trial with Saniona in mild and prodromal Alzheimer's disease. The PI of this grant is John Olichny, and the trial was meant to start last June, and unfortunately is on hold because of COVID. But because UC Davis had GMP material, we were able to provide tablets to Ulf Simonsen at the University of Aarhus for an open label phase 2 trial with Saniona in severe respiratory insufficiency associated with COVID-19 infections. And the last I've heard from Ulf is that he has met his enrollment target and is currently analyzing data. But as much as I like Saniona, it has suboptimal pharmacokinetic properties and no longer any IP. The patent expired in August 2019. So there really is a need for new KCS3.1 inhibitors with better ADME properties like SAN903 to go after any of these interesting indications. And with this, I would like to stop talking and thank you for your attention.
Thank you very much, Heike, for setting the stage in the ion channel field. Maybe we can move to the next slide. I'd also like to thank you for all your kind words about Saniona and about our ion channel team that has been among the leaders in the ion channel field for decades now. So I'm Jørgen Dreyer, the Chief Scientific Officer and the founder of Saniona, and I'll talk about what we call the Saniona ion channel drug discovery engine. Next slide, please. Some of Heike Buth's messages are also shared in this slide. She mentioned that ion channels as a drug target class is significantly untapped. It's actually only about 20% of the channels that have been successfully targeted today, and in general only with old and suboptimal drugs, and Saniona was an example of that. But still, it's estimated to be a $12 billion industry. As Heike mentioned, ion channels are complicated, and this is also illustrated in this cartoon. And that's why it has taken such a long time to master the ion channel drug discovery. They come in so many different forms and states, and until recently have been very difficult to crystallize. But ion channels can be mastered. And now I'd like to illustrate why Saniona is so well positioned to become successful in developing new effective treatments based on ion channel modulation. I'll talk about our compound library of more than 20,000 proprietary compounds and how this could lead us to the next generation of therapeutic candidate drugs. And I'll mention our proprietary database, which we call IONBASE. It's the core of our artificial intelligence efforts on making the next generation drugs. My colleagues would illustrate how our platform has delivered with SAN711 and SAN903 being the first two programs from our ion channel platform to enter into our proprietary pipeline. And I'll talk about how our platform is positioned to advance a continuous stream of new drug candidates. But first, we would like to show a short video to give you a real-life impression of our labs. So please put on the video. Thank you. And can I have the next slide, please? So Saniona is focused within rare diseases, and this gives a very good fit to treatment with ion channel modulators. Often these diseases are a consequence of mutations or in malfunctioning of the ion channels. So here I've listed some of the rare disease areas that are addressed by some of our most advanced ion channel programs. This is within the neurological conditions such as pain, epilepsies, movement disorders, hearing disorders. And Kai Nielsen will specifically talk about SAN711 in rare neuropathic diseases. We also address severe and rare inflammatory fibrotic conditions, and Pelle Christoffersen will cover this with a story on SAN903. And we also have programs in the field of, for instance, rare blood diseases, diseases in the genitourinary field, and even in rare cancers. So I think we can safely say that Saniona's ion channel programs broadly address many unmet medical needs in rare diseases. Next slide, please. This is an illustration on how we see our IONBASE database in the heart of our drug discovery efforts. It contains structural information on more than 120,000 compounds and the biological and technical data that we have generated on these compounds over the past more than 20 years. For each compound, it can be hundreds or even thousands of data points on how they modulate ion channel function. But it's much more than that, and I'll show that in the next slide, please. This is a cartoon illustrating all of the components of what we call our ion channel drug discovery engine, and also describing the iterative process of synthesizing, testing, and optimizing new drug candidates. Again, with IONBASE database in the center. All of these disciplines, including the in vivo studies, are in-house, and this gives us a tremendous advantage. I'll try to explain some of these steps on the following slides, and I'll start with discipline number 1 and 2. That's chemistry and precision biology. Next slide, please. So we start by making new molecules based on our existing knowledge and using parallel synthesis and molecular modeling. We then run the new molecules to a wide battery of dedicated cellular tests and ion channel binding and activity tests. And here we are taking advantage of our proprietary cell bank expressing specific ion channels. Next slide, please. I talked about the chemical library of around 120,000 compounds. You can see a snapshot of the library here and also in the video, and you can also see the high throughput robotics and screening technology that allows us to test thousands of compounds every day. In the blue box, we have listed some of the screening technologies that are used. So the compounds that show activity in the initial screen we call hits, and it's interesting to note that about 80% of the hits comes from this proprietary library of 20,000 compounds. And this speaks to the quality and enrichment of our proprietary library, and it explains how we can profit from repurposing proprietary compounds from the library into new programs. Next slide, please. So being an ion channel company means that electrophysiology is a very key discipline for us. And our electrophysiology team has for decades now been innovators in the ion channel field. For instance, with patch clamp automation, and they have published hundreds of articles in the field of ion channels. Here are some pictures from the lab showing some of the electrophysiology technologies. To the left, you see electrical recordings in frog eggs. And then there is a sophisticated patch clamp recording where we, in principle, can measure a single ion channel opening and closing. Then there is a in vitro brain slice recording setup where we can stimulate in one region and record the transmission of the signal to another region. And to the right, we show a robot that can perform several of these highly sophisticated recordings in parallel. Next slide, please. Like in all pharma projects, we need to know whether our new molecules are stable and how they distribute in the body. That's the box to the left, but also that they get to the target organ and hits the right targets there. That's the box to the right, which we call target engagement and translation. In the figure, we show an example on how a molecule taken by mouse is first absorbed, then distributed, metabolized, and ultimately excreted from the body. The green dots marks measurements of amount of drug in the blood, and the red dots are drug in the brain. So in this case, we could demonstrate that the molecule had a preference for going to the target organ, to the brain. Next slide, please. And finally, in vivo pharmacology and disease biology, where we demonstrate that the drug works in a real whole body disease setting. Saniona has specific expertise in psychiatry and neurology, in particular in epilepsy and pain. And many of the disease models we have developed ourselves, and we are able to show proof of concept in animals and assess pharmacological specificity and study adverse effect profile. Next slide, please. So all of this information is continuously fed into our IONBASE database on top of similar information on thousands of other molecules. And this is then used by our AI approach to refine our prediction models and aid designing the next even better molecule in an iterative process. You'll hear more about that later. Next, please. So to sum up, where are we going with this? We already have SAN711 going into the clinic now, and SAN903 should follow in about 1 year later. We have several drug discovery programs in the pipeline at different stages, which we call lead optimization stage, hit to lead stage, and even earlier programs still in validation phase. So we're confident that we'll be able to deliver a continual flow of new drug candidates to fill Saniona's pipeline. Next slide, and that is my last slide. I'll just show our proprietary pipeline slide where we see Tesomet in the top in late stage clinical development in 2 indications. But Tesomet is not the main focus for today. We would like to spend a little more time on SAN711 and SAN903 that are now moving up in our proprietary pipeline. And Palle Christoffersen and Karin Nielsen will join later to talk about these 2 programs. But first, I'll now leave the floor to David Brown, who is responsible for our computational and artificial intelligence efforts, and he will elaborate a bit more on how we use IONBASE. So please, David.
Thank you, Jørn. In the next few slides, I'll illustrate how we leverage our IONBASE resource together with AI and computational modeling to accelerate our R&D. So drug discovery runs in iterations. This is very much like any other inventive process. So prototypes are made. In our case, these are novel chemical entities. And these prototypes are tested and evaluated in each of the various disciplines to find out how well they've performed with respect to the project goals. Improvements are proposed, and these improvements are incorporated into a new set of prototypes. We cycle through this until we can tick off all of the project success criteria and propose a preclinical candidate. This process requires optimization of hundreds of parameters: the compound's activity on the target, the lack of activity on targets that can cause side effects or toxicity, as well as parameters describing what happens when the compound enters the body, druggability, and so on. So you can imagine that a vast amount of data is generated in the course of a project's lifetime, and these data can be challenging to keep track of. This is where computational modeling can support the project team. A model can be considered a summary of all the learnings that have been made based on the data. So actively using these models by calculating the expected data before compounds are synthesized ensures that this know-how isn't forgotten when designing the new compounds, and certainly that we don't waste time making things that we could have predicted will fail. Drug discovery requires finding the optimum balance of activity and safety and druggability. These properties are rarely independent of each other. You can improve one aspect but make another worse. So by combining predictive models for all of the most important assays, we can optimize new ideas across all of these assays simultaneously. An experienced medicinal chemist can perform this across maybe a few, but the reality is that we need assistance when it comes to so many parameters. So integrating modeling and AI into the drug discovery process can reduce the number of cycles that are necessary to go round from an initial idea or hit compound to identifying a novel compound that passes all the project criteria. In essence, we can save time and money by making the right compounds on the shortest path to a preclinical candidate. So how do we actually use this at Saniona? Here's an example from a current Saniona lead optimization program. This is a program in the final stage where only a little bit of fine tuning is required before we can nominate a candidate. The core structure of the prototype had been optimized in the earlier stages of the program. So this is the gray circle in the middle of the cartoon here. And we only needed to decorate this a little bit. There were 4 places on the core scaffold that we wanted to investigate, and around 20 standard chemical groups that we selected to try on each site. When you do the maths, you can see that even in this fairly limited fine-tuning scenario, there are 160,000 possible compounds. It would have been an enormous and expensive undertaking to make and test each of these. So instead, we extracted the curated data for the most important assays from IONBASE, data already generated in the program during the earlier stages. Then, using AI and other techniques, we built computational models for the key assays and combined these to predict the data profile of each of these 160,000 compounds in silico. We then prioritized the possible compounds based on the chance of success across all of these parameters that we had modeled. We threw away the ideas with very low probability of making it to the clinic, and we only made physical samples of the ones with the best chance, the ones that matched the candidate's selection criteria the best. These compounds were tested, and many have moved on to the next level of testing. In addition, the data from the first round was entered back into IONBASE so that all models could be refined and a new set of compounds made. In this way, the project has been able to home in on the few compounds that will pass all of the project selection criteria. On this slide, I'd like to explain why IONBASE is so important to Saniona's R&D. Data are key, regardless of the type of computational modeling you wish to run. Computational methods such as AI and machine learning algorithms are powerful tools that can be applied to the data, but they are in essence just powerful mathematical tools. In order to learn, these tools need to be trained, and it's impossible to train AI without data, no matter how good your platform is. We can mine the literature for data, and many do this, but with IONBASE, we already have an extensive data set at our fingertips, data for thousands of ligands tested in hundreds of assays, all of the parameters that we need, ion channel activities, as well as general druggability and pharmacokinetics. Without these data, without IONBASE, it would be much more challenging for us to generate relevant models to assist our ion channel drug discovery process. So IONBASE is Saniona's core competitive advantage. So my final slide, in summary, with IONBASE as the foundation and the heart of Saniona, and integrating AI and computational modeling into all areas of our research, we're able to drive the drug process faster, and so meet our goal of identifying one new preclinical candidate every year. Thank you for listening, and now I'll hand over to our Senior Vice President, Pelle Christoffersen, who will talk about SAN903.
Thank you very much, David. I'm Pelle Christoffersen. I'm Senior Vice President of Research, and I'm leading a group of ion channel specialists and highly skilled and experienced research assistants here at the Danish side of Saniona. The responsibility of that group is to set up in vitro channel assays to screen and characterize compounds from our proprietary compound library and to establish the effect of compounds on important cell types such as neurons, microglia cells, fibroblasts, smooth muscle cells, and erythrocytes, etc., or complete tissues such as brain slices and muscle strips. And now I'll be happy to give you a brief overview of SAN903. Next slide, please. SAN903 is an inhibitor of KCA3.1, and this notion stands for a specific potassium channel which is activated by small increases in intracellular calcium. SAN903 thus shares the molecular target with Saniona, the compound that Heike Wulff mentioned in her talk previously. However, in contrast to Saniona, SAN903 is a next-generation compound with a long patent lifetime, and it comes with a greatly improved physiochemical properties and also we have evidence of an alternative mechanism of action on this channel, which we think may be beneficial. So SAN903 can therefore become a first-in-class registered medicine based on inhibition of this channel. The status is that SAN903 is in preclinical development, and we expect the phase 1 studies to begin mid-2022. KCA3.1 is expressed in certain immune cells and fibroblasts, as well as in erythrocytes and even in some cancer cells. And as illustrated on the cartoon to the right, inhibition of this channel indirectly leads to a dampening of the influx of calcium from the outside to the inside of these cells. And since calcium is such an important signaling molecule for activation of intercellular processes, including specific gene regulations, inhibition of this channel can inhibit pro-inflammatory cytokines from immune cells and excessive collagen production from fibroblasts, and it can also inhibit proliferation and cell migration of these cells. Next slide, please. I'll now show here briefly one example of how SAN903 may work at the cellular level. We have performed patch clamp electrophysiology, one of the key ion channel technologies that Heike Wulff mentioned in her talk. We have here used the technology on a fibroblast, which is derived from a patient suffering from the serious lung disease idiopathic pulmonary fibrosis, often just abbreviated to IPF. What we measure here is actually the membrane potential of this fibroblast. The membrane potential is the difference between the voltage in the extracellular phase and the voltage in the cell's cytoplasm. When we experimentally activate the channel, which is here shown as the yellow part of the figure, the membrane potential immediately becomes very negative, which simply reflects that positively charged potassium ions are now passing through the open channel from the inside to the outside, leaving the negative ions behind. And this point is actually essential, since it is exactly this strongly negative membrane potential that drives the double-charged positive calcium ions from the outside into the cell, as shown in this cartoon on the right side. Application of SAN903, which is here shown in the blue part of the figure, we are now back in the left side, inhibits the potassium channel, and quickly the membrane potential returns to the low value, where the calcium influx becomes small and ineffective for regulating intercellular signaling. And now to the involvement of the channel in various diseases. Next slide, please. Due to the expression of the channel in certain immune cells, it can play a pathological role in overactive immune responses in some chronic inflammatory diseases. And likewise, Due to its presence in activated fibroblasts, it can play a pathological role in chronic fibrotic diseases such as idiopathic pulmonary fibrosis. Finally, I will also mention here that it is expressed on erythrocyte membranes and that it plays a pathological role in certain rare blood diseases such as sickle cell disease and hereditary steatosis. And in a subset of the latter disease, mutations in the channel gene are even what directly causes the disease. And then I'll now show a few examples of how SAN903 works in models of diseases. Next slide, please. The first example is a model of intestinal inflammation, which is, for example, a characteristic of inflammatory bowel diseases and also rare complications to some of these diseases. What you see here in the photograph in the left upper corner is 4 severely inflamed colons from rats, and the arrows mark up damaged and necrotic areas known as ulcers. In the photograph below, you see similar colons from animals that have now been treated with SAN903 for 1 week. And from a visual inspection, it's clear that the ulcers have largely disappeared and that the swelling of the tissue is also remarkably decreased. On the bar graph to the right is shown a quantification of the healing effect of SAN903 obtained at different daily doses in comparison to sulfasalazine, a medicine for maintenance treatment of inflammatory bowel diseases. Obviously, as seen from the x-axis of this graph, SAN903 is much more potent than sulfasalazine, and importantly, it is also as effective and nearly as potent as steroids, the class of anti-inflammatory medicines that can only be taken at a limited time span and therefore only are good medicines for treatment of acute flare-ups of the diseases. Next slide, please. The second example that I will show is a model of idiopathic lung fibrosis. What you see here is the top-line results from a presentation by my colleague, Deepak Amrutkar, at the recent Experimental Biology Conference. In this study, we actually compared SAN903 head-to-head with 2 registered medicines for idiopathic lung fibrosis, nintedanib and pirfenidone, administered close to the equivalent human therapeutic doses. As seen from the 2 top bar graphs on the right, looking at lung histology, the conclusion was that SAN903 had better effects than both of the medicines with respect to dampening of the general infiltration, as well as to deposition of collagen in the lung tissue. It is noteworthy, however, that these positive effects were not directly related to a depression of the transforming growth factor beta-1, which was markedly and statistically significantly reduced by nintedanib in this study, despite this medicine's lack of effect on the histology part of the model. In my opinion, this clearly indicates the different mode of actions of SAN903 and nintedanib, and probably also the different levels of intervention with the disease cascades, which one may speculate at least may tentatively rationalize a possible combination therapy in the clinic. Next slide, please. In this, I will just sum up this very brief review on SAN903 by stating the few main points of the talk. SAN903 is a next-generation inhibitor of KCA3.1, a calcium-activated potassium channel. SAN903 has shown promising efficacy in vitro and in vivo models of inflammation, fibrosis, and also in blood disorders. Specifically, SAN903 showed a better effect in lung pathology in a model of idiopathic lung fibrosis than the 2 already marketed products, which may indicate that it can become a potential first-in-class and best-in-class medicine. Lastly, I will just repeat that SAN903 is in preclinical development and that the phase 1 study in healthy human volunteers is expected to begin mid-2022. Thank you very much, and I will now hand the slide deck over to Karin Sandager-Nielsen, our Vice President of In Vivo and Translational Pharmacology, who will present SAN711. Thank you.
Thank you very much, Palle. My name is Karin Sandager-Nielsen. As he said, I'm Vice President of In Vivo and Translational Pharmacology, and I'm the scientific lead on the SAN711 program. The focus of my talk will be a review of some of the preclinical data we have generated on this molecule. Next slide, please. SAN711 is a first-in-class positive allosteric modulator of GABA-A alpha-3 receptors. The target indication is rare neuropathic disorders, and it's entering phase 1 study in healthy volunteers in mid-2021. And the data from this study is expected by early 2022. As stated, SAN711 is a highly selective modulator of GABA alpha-3 receptors in the spinal cord. This is a very unique profile, and to the best of our knowledge, it's actually the first of its kind. And it's specifically designed to restore dysfunctional spinal inhibition associated with neuropathic disorders. And in the following slides, I'll unpack this mechanism a bit more and present some of the data generated to support the indication. Next slide, please. The molecular target of SAN711 is the GABA-A receptor. And the GABA-A receptor is a ligand-gated ion channel that is activated by GABA. And GABA is the main inhibitory transmitter in the brain. And when it binds to the receptor, the channel opens and chloride ions flows into the neuron. That hyperpolarizes the neurons and inhibits neuronal activity. SAN711 binds to a site distant from the site where GABA binds on the receptor. And that allows SAN711 to modulate the effect of GABA. The channel consists of 5 subunits with various subtypes, typically 2 alpha, 2 beta, and 1 gamma subunit. And the function of the receptor complex is determined by the subunit composition. Using mice with genetic modifications in the different alpha subunits has shown that activation of alpha-1 receptors is associated with sedation, motoric instability, abuse, and dependence liability, while activation of alpha-5 receptors is associated with cognitive impairment. These are all very well-known side effects shared by a group of drugs called the benzodiazepines. And the benzodiazepines non-selectively activates GABA-A receptors containing an alpha-1, 2, 3, and 5 subunit. And SAN711 critically differentiates from this group of drugs in that it selectively enhances the effect of GABA on alpha-3 containing receptors only. Next slide, please. And this slide exactly shows you the unique functional efficacy profile of SAN711. The y-axis represents the GABA-activated chloride influx, while the x-axis represents increasing concentrations of SAN711. And what is evident by inspecting this figure is that SAN711 only potentiates GABA's effect in cells expressing alpha-3 receptors, which is the blue line, while there's no or very low effects on cells expressing alpha-1 and alpha-5 receptors, as seen by the red line and the yellowish line respectively. And as I stated before, this is a very unique profile, and it's predicted to retain all the beneficial effects of enhancing GABA at the spinal cord level, while at the same time effectively avoid the adverse effects mediated by activating alpha-1 and alpha-5 receptors in the brain. Next slide, please. And GABA-A receptors containing an alpha-3 subunit are not very prevalent. In fact, they only constitute around 10% of the entire GABA-A receptor population with this binding site. And this is a good thing, as you want your target to be as specific as possible. And furthermore, it's localized in the right areas to affect pain transmission, namely at the spinal cord dorsal horn, lamina 2, which is where the primary efferent fibers that carries the nociceptive information synapse with the secondary neurons, which transmits the signal up to the brain. So from an anatomical point of view, it is localized in a perfect position to modulate and reduce nociceptive transmission in the pain pathway. And strong analgesic effects of SAN711 have been confirmed in our rodent studies, which I will show you here in the next slides. Next slide, please. So, firstly, we've used a rat model for trigeminal neuralgia, which is a rare neuropathic disorder with a great unmet need. Current standard of care is the sodium channel blocker carbamazepine, which is very poorly tolerated and associated with numerous adverse effects. If you look at the figure, the y-axis represents the pain threshold, so increasing numbers means less pain, while the x-axis represents the different phases of the study. And what is clear by inspecting the figure is that SAN711 results in a very clear and dose-related analgesic effect with a minimal effective dose of 1 mg per kg. And 1 mg per kg results in a plasma level giving rise to approximately 50% receptor occupancy. Furthermore, the highest tested dose, the 10 mg per kg group, which is the dark blue line, has an effect size similar to carbamazepine, and this dose results in a full reversal of pain behaviors, meaning that these rats behave completely normal as if they had not been stimulated the day before. Next slide, please. So this study is actually an extension of the previous one, because in this study, we wanted to investigate if repeated treatment with SAN711 prior to the stimulation is able to prevent the development of pain. And secondly, if this repeated treatment would result in development of tolerance, because tolerance development is one of the critical aspects that prevent the chronic use of benzodiazepines. So the design of this study is basically similar to the one on the previous slide, except for the fact that the rats are being dosed repeatedly with SAN711 prior to the stimulation, and then again immediately after the measurement of hypersensitivity at the 0 time point. And this is reflected by the blue shaded areas. So that means that the pain readouts at the 0 time point is performed drug-free, and this is important. Because what is remarkable here is that in contrast to rats treated chronically with vehicle, and that's the black line, who all show a marked hypersensitivity at this time point, rats treated chronically with SAN711 at 1 and 3 mg/kg, which is the blue and green line, all show significantly reduced pain behaviors. And as I said before, as they are tested drug-free, this can only mean that the chronic treatment prior to the stimulation has prevented the development of pain. Of course, it's important to note that chronic administration is not associated with increased exposure, so the data cannot be explained by accumulation of the drug. Furthermore, chronic administration prior to the stimulation results in more potent analgesic effects, and that's evidenced by reduction of the minimal effective dose down to 0.3 mg/kg in this study, which is the light blue line, as compared to 1 mg/kg after acute treatment. Moreover, a full reversal of pain behaviors is now attained at 3 mg/kg, which is the green line, as compared to 10 mg/kg after acute treatment. Lastly, and very, very importantly, the analgesic effect is maintained after repeated dosing, showing that chronic administration of SAN711 does not lead to tolerance development. And this point is further supported by the data on the next slide. So next slide, please. So this is the last set of pain data that I'll be showing you. And the purpose of this study is twofold. Firstly, we wanted to reproduce the effects of SAN711 in another standard rat model of neuropathic pain, namely the chronic constriction injury model. Secondly, we wanted to further address the lack of tolerance development that we also saw in the previous study. And for that purpose, we included morphine as a positive control, as it's well known that strong tolerance develops after repeated treatment with this opioid. Again, the y-axis represents the pain threshold, and the x-axis represents the different phases of the study. Firstly, development of hypersensitivity is confirmed by drug-free baseline measurement. After that, the rats are treated with increasing doses of SAN711 and morphine, and the analgesic effect is assessed. Then we continue dosing for 7 days, whereafter the pain threshold is measured again. And the main conclusions from this study are that acute treatment with SAN711 and morphine, for that matter, results in significant analgesic effects, as evidenced by enhanced pain thresholds on treatment day 1. After 7 days of continued treatment, the analgesic effect of SAN711 is fully maintained, while the effect of morphine is completely lost, as shown by the red arrow. And this strongly supports that SAN711 is highly effective in an animal model for nerve injury, and that repeated treatment is devoid of tolerance development. Next slide, please. So, whereas the last 3 slides have confirmed the analgesic activity of SAN711, this slide addresses if this unique subtype selective profile actually translates into lack of adverse effects typically associated with GABA-A activating compounds. And one of the dose-limiting adverse effects of the non-selective benzodiazepines is strong sedation. Sedation in rodents is easily monitored by placing them into a novel environment. Non-sedated rats will spend their time actively exploring, while rats dosed with, for instance, diazepam, as you see on the left figure, will explore less as they are sedated. In contrast to this, rats dosed with even very high doses of SAN711, resulting in full receptor occupancy, as you see on the right-hand figure, fully maintains the ability to actively explore to the same extent as control rats. And these data convincingly show that SAN711 does not lead to the strong sedative effects shared by other GABA-A activators, such as the benzodiazepine. And they further confirm that the unique subtype selectivity of the molecule that we have designed truly does translate into a predicted and desired in vivo profile. Next slide, please. So this is my last slide. In summary, SAN711 represents a novel first-in-class approach. It selectively enhances the effect of GABA on alpha-3 containing receptors. And this profile is designed to be ideal for treatment of rare diseases associated with neuropathic pain or itch. It's devoid of the typical adverse effects associated with the non-selective GABA-A activation, and we have a strong preclinical proof of concept in vivo models for neuropathic pain. Further, the effects in these models are maintained after chronic treatment, indicating that of tolerance development. And we are very excited about this molecule and the mechanism, and it's entering phase 1 in healthy volunteers in mid-2021. Data are expected by early 2022. And with that, I'll leave the floor to our Chief Medical Officer, Rudy Baumgartner, to inform you on the clinical aspects of this program. Thank you.
Thank you very much, Karen, for this excellent presentation. I'm Rudolf Baumgartner, the CMO for Saniona. It's my pleasure to be here. As discussed so eloquently by Karen, SAN711 is a very selective GABA alpha 3 modulator with an enormous potential for the treatment of neuropathic pain. In this slide, you see a schematic for the upcoming SAN711 phase 1 trial. The trial has several objectives. The primary objective is to determine the tolerability and the MTD, or maximally tolerated dose, for SAN711. A secondary objective is to determine drug localization, or where in the body the drug goes to, and importantly, at what level of receptor occupancy. As Karen discussed previously, receptor occupancy is associated with efficacy at the receptor level. The study will involve approximately 80 to 88 healthy volunteers. Subjects will be randomized to SAN711 or placebo. It will have 3 phases: a single ascending dose phase, a PET evaluation phase, and a multiple ascending dose phase. And this trial is scheduled to start mid-year. So we're pretty excited about this. As a reminder, Saniona is conducting clinical trials with Tesomet as well. And Tesomet, just for us to know, is a fixed dose combination of tesofensine, which is a triple monoamine reuptake inhibitor, and metoprolol, which is a selective beta-1 blocker. Tesomet is targeted for hypothalamic obesity, or HO, and PWS, which is Prader-Willi syndrome. HO, just to remind us, is an acquired rare disease that can be developed after hypothalamic injury, such as a consequence of brain surgery or radiation following craniopharyngioma. And HO is characterized by severe intractable weight gain, and it's not unusual for patients to double in size within 12 months of onset. Currently, there's no approved treatments for hypothalamic obesity. PWS is a genetic rare disease that is characterized, amongst other things, by severe hyperphagia or the inability to achieve satiety. There's also currently no approved treatment for PWS other than growth hormone, which does not treat the severe hyperphagia, which is pathognomonic PWS. The triple monoamine reuptake inhibitor, tesofensine, increases the levels of dopamine, serotonin, and noradrenaline in a very balanced manner and with a long and steady half-life. This results in a clinical reduction in food craving, appetite reduction, and increases in metabolic rate. However, because noradrenaline, in addition to increasing metabolic rate, can also cause increases in heart rate, and at higher doses than in our clinical program, it can also induce sporadic changes in blood pressure. Metoprolol was added to this triple monoamine reuptake inhibitor to create a cardiacally neutral drug, and this drug is Tesomet. These are recent updates within the 2 clinical programs. We've received from the FDA orphan drug designation for Tesomet and PWS, and this confers on us several benefits. One, it reduces our fees. it can grant us orphan protection afterwards from a patent perspective. But most importantly, what it does for us is that it allows us to communicate with the division more frequently, and which is very important for us. We also opened the investigational new drug application, or the IND, for PWS with the FDA. We've received further clarity from the FDA on the regulatory pathway for hypothalamic obesity. We've selected our clinical research organizations, and we are currently selecting trial sites for both phase 2b studies in PWS and HO. We selected our contract manufacturer for our phase 2b and phase 3 trials in PWS and HO. These are the outfits that make our drug and supply it. And we are currently collaborating with PWS and HO patient advocacy organizations. Part of that effort is to get patient input as well as caregiver input to optimize our trials, and as well as communicate to them that these trials are about to happen. This is a schematic of the upcoming phase 2b trial in PWS. It's a double-blind, randomized, placebo-controlled trial of roughly 110 to 120 subjects to be conducted in the US and ex-US. It will study placebo and 3 doses of Tesomet. The double-blind portion will be about 4 months in duration, followed by an open label period. The trial is scheduled to start the second half of this year. The next slide is a schematic for the proposed phase 2b HO trial. It is a double-blind, randomized, and placebo-controlled trial of roughly 120 subjects, also to be conducted in the US and ex-US. It will study placebo as well as 3 doses of Tesomet. The double-blind portion will be about 9 months in duration, followed by an open label period. This trial is also scheduled to start the second half of this year. Anyway, thank you for your time. And now I would like to hand this presentation off to Rami, our CEO, for concluding remarks.
Thank you very much, Rudy. And I'd just like to maybe walk through some of our key milestones over the next year and a half. So during the first quarter, we have announced, and as Rudy mentioned, we received orphan drug designation for Tesomet in PWS. We are pursuing orphan drug designation for HO as well. We have also aligned on the regulatory path forward for Tesomet in hypothalamic obesity. And as I mentioned earlier, our next clinical trial will be indeed a phase 2b, which we plan to initiate in the second half of this year. Later this half, over the next couple of months, we plan to initiate our phase 1 trial with SAN711, as we mentioned earlier. And later in the year, we will initiate the 2 phase 2b trials, both for PWS and for HO. As we move into the first half of 2022, we plan to have the data available from the phase 1 trial from SAN711, as well as initiate the phase 1 trial with SAN903. And as we move to the second half of next year, we plan to have the data from the 2 phase 2b trials towards the end of the year, and initiate the phase 2 trial with SAN711. And this schematic is really just to illustrate that, we're excited about Tesomet, obviously in mid to late stage clinical development, but Tesomet really is only the beginning for us. We plan to build a fully integrated company and be a leader in rare diseases, starting indeed with Tesomet in HO and PWS, but that will then follow with SAN711 for rare neuropathic disorders. It will then follow with SAN903 for rare inflammatory disorders. We will then explore our 20,000 ion channel modifiers with the goal of identifying new treatments for new rare diseases. We will then explore new indications for the treatments that we have through life cycle management, being Tesomet, SAN711, or SAN903. And we will also look externally for business development opportunities to bring into the company that would be a good fit as we grow our company. And with that, I would like to thank all the presenters. I'd like to thank all of you for your attention. I'd like to pass it back to the operator to open up the Q&A session. Thank you very much.
Thank you, Rami. At this time, we'll be conducting our Q&A session. As a reminder to the audience, you may submit your questions through the Q&A text box at the bottom of the webcast player or by emailing them to questions@lifesciadvisors.com. So you may go ahead and post your questions, and please hold while we pull for questions, and then I will turn it over to Trista at Saniona to moderate the Q&A.
Okay, thank you everyone for joining us today. We will kick off our Q&A now. And again, if you have questions, please feel free to submit them online. Our first question comes from Elisa Bako at Evercore ISI. She asks, can the phase 2B trial that you're planning for Tesomet in HO be registration enabling, or do you anticipate that you will need to do a phase 3 trial following it? So I will kick that one over to Rudy.
It's a great question. So currently we have a pre-IND meeting, which we have an understanding from the pre-IND meeting that they requested a dose ranging trial followed by phase 3. However, you always know that, we've conducted a trial in Denmark where the alpha or the p-value was extremely high, showing efficacy in hypothalamic obesity. And it's conceivable, depending on what the data shows in phase 2B, that there may be conversations with the agency. But at the moment, our plan is to phase 2B followed with an end of phase 2 meeting and the phase 3.
Excellent. Thank you, Rudy. So our next question is from Anders Hedlund of Redeye, and he's asking about the clinical trial that we are planning for SAN711. He asks if that will take place in the U.S. So Rudy, I'll toss that to you again. Sure.
The SAN711 trials will be conducted in Europe, mostly because it was facilitated by, we found a site that was near a pet center who have run these types of studies in the past. So for a efficiency perspective, it's going to be conducted mostly in the UK. So after that, our plan is to transfer the phase 2 trial that will follow to the US. Thanks.
Thank you, Rudy. A related question to that on SAN711. Why has it taken SAN711 so long to get into the clinic, and what has sort of been the process to get SAN711 ready for the clinic?
So to get SAN711 into the clinic required us to complete what's called a pre-IND package, which requires a lot of toxicology. It's something you start from scratch. You also have to align with the regulatory agencies that the dosage you studied in animals are adequate. So all that conversation has been ongoing, and it takes months, frankly. You submit the package, they review it, it comes back, it goes to institutional review boards, they basically review your study. Because this phase 1 study is not as simple as some of them, it's taking a little bit more back and forth, but we're pretty excited. The study has been approved by an IRB and we're ready to go. Thanks.
Excellent. Our next question comes from Jay Olson at Oppenheimer. Jay asks, is there a potential risk of tachyphylaxis when targeting ion channels with chronic dosing? And how can drugs be designed to avoid tachyphylaxis? So perhaps Jürgen, Karen, and Heike, you all might have some input on that.
Yeah, I could start by saying, yeah, clearly there is potential for that you lose the effect with time. You have tolerance development, tachyphylaxis with ion channel modulators. So that's why you need to be very careful and go for specific subtypes. And I think Karin showed very nicely that we are actually able to avoid tolerance development, avoid tachyphylaxis with the treatment of SAN711. on GABA, on addressing specifically GABA alpha 3. So I think it is quite an interesting opportunity rather than an obstacle that we can avoid tachyphylaxis, which we will see definitely with the opiates. Heike, maybe you would like to.
Yeah, I mean, I don't have very much to add because you already said it. I mean, for some channels, you see tachyphylaxis, you see, especially with GABA, benzodiazepines are really known for losing efficacy, which is why a lot of people go after extrasynaptic GABA-A receptors. So I was really impressed by your data showing that you're not getting tachyphylaxis. With other channels, you absolutely don't see it. So with sodium channel blockers, you never get tolerance. So this is something that needs to be studied.
Karin, do you have anything to add?
Oh, I could add that it's not known what causes the tachyphylaxis by the benzodiazepine, but most studies point to activation of alpha-1 receptors being involved. So clearly, we are avoiding activity of those, so that's helpful at least, and probably why we don't see development of tolerance.
Right, thank you guys very much. Actually, a follow-up question from Jay Olson at Oppenheimer. He asked, what is the target product profile for SAN711? And can we give any color on the initial indication and potential market opportunity?
So
Rudy, go ahead.
Yeah, so it's a great question. We are currently looking at neuropathic pain, and neuropathic pain, as you well know, it has several entities underneath it. Some of them are rare, some of them are common. So the data is pretty strong for trigeminal neuralgia, and there's other indications there as well. So we haven't really declared where we're going to go. I mean, it's something that the company is working on. We have a pretty good idea. It hasn't been made official yet of where we will be, but it'll be under the umbrella of neuropathic pain. And then there's a couple additional criteria that we're trying to meet. The phase 1 study that will be conducted is relevant because not only will it tell you that the drug is safe and well tolerated, and as Karen mentioned, it's devoid of alpha-1 agonism, which would mitigate sedation. It doesn't have an alpha-5 activity, which would mitigate cognitive dysfunction. So we would get that profile from the phase 1, but the PET scan part of the study is really interesting because what it allows us to do is understand at what levels each of the doses that we will test will occupy the receptor. And based on that, we'll get a really good risk-benefit profile or an understanding of what it should be. Based on that, we will know whether we can go for very focused diseases or whether we can broaden it out. So it's something that we should be communicating to the street within a few months. Thanks.
Thank you, Rudy. Our next question comes from Gary Nachman of BMO Capital Markets. Talk about the capacity that the R&D organization has to continue to expand the clinical programs in the pipeline and as you try to further build out the rare disease portfolio. Do you have the capabilities that you need in-house, or do you anticipate that you are going to need to bring in new technologies or capabilities over time? Rami, I don't know if you want to weigh in on that, and then perhaps Jürgen and Rudy can add.
Sure, I'm happy to start. So maybe just as a reminder, just a year and a half ago, we were only a research organization. We had no clinical development capabilities. We have built those clinical capabilities over the past year and a half or so. We now have a very strong research organization, as you heard today, and we have a very strong clinical development team under Rudy's leadership. We have what we need to run what we are currently planning to do. But as we plan to grow, as we plan to bring in new indications and new molecules and new treatments, We will, of course, need to continue to grow accordingly to make sure that we have, the sufficient experience, capabilities, and manpower, quite frankly, to be able to do all that we want. But for now, for what we are currently doing and what we talked about now, we do have the capabilities. Rudy and Jürgen, is there anything you want to add?
Sure. From a model, the way the clinical development program or the clinical development team, which frankly goes to Saniona as a whole, is almost like a hybrid program. So we're hiring individuals who have 20 to 25 years experience in each of the functions. So the purpose of that model is for them to be able to manage outside organizations and provide basically experience and leadership when they run into problems. These individuals, and the way that the system is set up, is that we would be able to manage multiple programs at a time. So it's not like, we have these sort of the very, very experienced individuals in each of these functions, and then you can put many different programs underneath them. There is a capacity, of course, the company needs to grow, but it's being built to be able to manage multiple programs. And the hope is, and Jürgen can talk about this, is that the research engine, which is certainly there, can provide new clinical assets at a rate of at least a new clinical asset that enters the clinic with good probabilities of success, at least one molecule a year, which is not inconsequential.
Yeah, I think the question was mainly about how we would make the transition from the preclinical into the clinic and actually move the compounds to the clinic. As Rami started, we do have the research organization fully built up. We worked together for many years, and we tried to illustrate today how we have not only an engine, but also a pipeline of earlier stage programs in research that should be able to feed our pipeline for some years to come. But of course, we are also thinking out of the box, thinking into new modalities to add on our platform. Also, David talked about our artificial intelligence efforts on our IONBASE. So we are also really developing our research organization to be also competitive and innovative in the future.
Right. Thank you guys very much for all weighing in on that. Our next question comes from Anders Hedlund at Redeye. Again, he asks, this question is for Rudy. Can you give us some flavor on your interactions with the FDA in the wake of their efforts in fighting the COVID-19 pandemic? Are you feeling like you're getting airtime with the FDA, and are they able to respond to you in a timely manner, or have you seen difficulties due to the pandemic?
Well, it depends on the division, frankly. We basically, the divisions that we're currently working with, have been as responsive as before, which is psych division and the division on metabolism and lipids. We did, when we were looking initially at SAN711, perhaps started in the US, one of the reasons we decided to remain in Europe is because we were informed there was a backlog at the FDA due to COVID-19. So I mean, it depends on the division and their capacity. So currently, for the two programs that we have in the US, we have not felt it. We just got sort of a little bit of a signal that maybe if we had opened SAN711 in the US, we may have been affected somewhat by COVID-19.
Very good. Our next question. So a lot of the presentation today focused on IONBASE, and what about IONBASE is truly proprietary or unique to Saniona? So Jürgen, perhaps you could take that one.
Yeah, as I mentioned, the IONBASE has been developed over more than 20 years, and it's based on input from more than 120,000 compounds. So all of these data are proprietary, and the 20,000 of the 120,000 compounds are proprietary compounds. So also these compounds are proprietary. So that it is really a very unique platform that we've been able to develop over, and it takes a long time. All of these programs have been ion channel programs, so it's also optimized in that field, and that's why also we get such a high hit rate when we screen these compounds on other targets or other ion channel targets. So it is, I think, very unique. And as David mentioned, this also means that we can apply an artificial intelligence approach on this database and get really meaningful predictions out of it, which can be otherwise impossible if you don't have a strong database.
Thank you, Jürgen. Another question here for Heike. You touched on the importance of achieving selectivity as a trait across ion channel modulators. How do you think this is going to play a role in the future of drug development? And do you see a lot of excitement for ion channel modulators in the future?
Absolutely. I mean, the families where it's difficult to get selectivity are, of course, the sodium channels, the KB1 family channels, GABA, depends on where your binding site is. It's very hard in the pore. It's easier in the extracellular domain. So I think this field is going to really open up as we're getting crystal structures, as people are developing artificial intelligence. So I think we're going to see many more ion channel targeted drugs going even after channels with very close homologs.
Thank you. Jürgen, is there anything you want to add there, or?
No, I would strongly support that.
Thank you. Okay, we have a question here from Adam Carlson at ABG Sundal Collier. He asks, based on the graph of subunit selectivity that you showed, are you confident that SAN711 is sufficiently selective for the GABA alpha 3 subunit and that it will avoid the other alpha subunits and the adverse effects that they are associated with? Jürgen, do you want to start with that, and then perhaps Karen can weigh in as well?
Sure. So the important subunit that you want to avoid is the GABA alpha 1, because that is, as Karen mentioned, what gives sedation, muscle incoordination, and many other side effects. And we are completely blank on alpha 1. There's no efficacy there whatsoever. I think that is really a major accomplishment in itself. There are a few other companies that have succeeded in getting rid of most of the alpha 1. But another subunit that we wanted to take down is the GABA alpha 5, because if you have a high efficacy on GABA alpha 5, then you are likely to get cognitive impairment. And that has been seen in animal models, but that has actually also been seen in humans. And this we also wanted to avoid. And we've got that down very, very significantly from, and also from competitor compounds. Nobody else have been able to make a GABA alpha 3 selective compound as this one, as Karen also mentioned. Karen, do you want to add?
No, I think you answered that very clearly.
Okay, excellent. Thank you guys very much. Here's another question along the same lines here. How is SAN711 different from Cerevel's darigabat?
Yeah, if we compare directly these two compounds, they have the similarity that they are very low on GABA alpha 1. Our compound is actually completely devoid of alpha 1. So that there we can say it is a selective compound in both cases. But in the Cerevel compound, they've not managed to get the alpha 5 down. And maybe they are going for other indications where this could be a good thing to have but we don't want to have it when we go for the neuropathic pain conditions and the other disease areas that we have focused on. So that is the main difference, alpha 5.
Great. Karen, anything you want to add there? Okay, great. Thank you. So switching gears a little bit over to the clinical side, a question for Rudy. Will the Tesomet studies in HO and PWS start when they are fully recruited, or do they start and then recruitment is ongoing? Maybe you could just give a quick primer on the clinical trial process there.
Right. So the way the clinical trials begin is basically you submit a protocol proposal to the FDA, and the FDA has a set number of days to get back to you, depending, it's 30 or 60 days, depending if the IND is open. You get back once you get the okay from the agency, you then open the clinical sites, and typically the clinical sites are open. You deliver drugs in blocks to the clinical sites, and the clinical sites are open, and then recruitment begins. And they screen patients, and the patients who are screened that meet eligible criteria, because there's inclusion exclusion criteria, then proceed. And those patients then that are eligible get randomized. This randomization process is on an ongoing rolling basis. So in general, these clinical trials, probably will be in the range of 8 to 12 months to recruit them all, meaning for the last patient to be recruited. So things that we look at are first patient in, last patient out, and when top line data is available. Those are the big milestones that's where we plan our activities around.
Thank you, Rudy. A question here, perhaps for Rami, also about Tesomet, actually about tesofensine. Can you comment on the status of the approval of tesofensine in Mexico?
Sure, thank you for the question. Yes, as some of you may know, the file was submitted for approval in 2019. Initially, they were expecting approval in the second half of 2020. Unfortunately, COVID hit, and it really hit quite strongly in Mexico, where the Mexican authorities stopped reviewing any files and only focused on COVID-related matters. Towards the end of 2020, they refocused again on reviewing files across all therapeutic areas. We have been informed by our partner medics that they have or are reviewing tesofensine now. They've started already. And what we were informed is that they are expecting an approval around mid-year. So it is imminent, but we are solely dependent on the Mexican authorities to finish the review and ultimately, hopefully, approve it. The expectation is around mid-year.
Okay, thank you, Rami. So I'm getting told that we're running up to the end of our allotted time here. So we'll take one more question here. And if we did not get to your questions, please feel free to reach out to me directly. All of my contact information is on the Saniona website under the investors section. So again, if you have any questions we didn't get to, you can send them my way. So Anders Hedlund at Redeye, one more question here. This is about some of the earlier stage ion channel programs that Jürgen and the team are working on. Specifically, he asks about the GABA program that we regained from Boehringer, and if we are currently working on that program as well.
Yeah, I can confirm that we are definitely working on that program. It was a very high quality program that we got back from Boehringer. They changed strategy and left it back with us. It's several hundred of compounds that are very potent on this specific target called GABA alpha 5. And we've been evaluating which indications would be best for us to move into. And we have not published any on that, so I cannot go deeper into that. But definitely we see this as a very, very promising program.
Excellent. Okay. Thank you guys so much. Thank you, Saniona team, for your time and your work on these presentations. And thank you everyone who dialed in and joined us today. We really appreciate your interest in our ion channel pipeline. We're super excited about it, and we hope that you are as well after everything you've heard today. And a special thank you to Dr. Heike Wolf for waking up early California time and joining us today to share her ion channel expertise. So again, thank you everyone, and feel free to reach out directly with any additional questions. Have a great day.